Thank you for showing interest in my research. My name is Takudzwa Madzivanzira. I'm a final year PhD student based at the South African Institute for Aquatic Biodiversity, SAIAB, and I'm registered with Rhodes University in South Africa. My poster presents a study that we conducted in Lake Kariba, Zimbabwe, to compare two methods that were used in Southern Africa for the invasive Australian Redclaw crayfish abundance studies in order to determine the effective gear that can be used for population suppression and to determine a standard gear for future crayfish abundance studies. We assessed for differences in detection probability and catch per unit effort of opera traps baited with cooked maize meal used for crayfish surveys in Zimbabwe compared to collapsible proma traps baited with dry dog food used for crayfish surveys in South Africa, Swaziland, and Zambia. Detection probability and catch per unit effort were significantly lower for opera traps compared to proma traps. The observed significant differences in catch per unit effort between the two methods could be attributed to either the difference in trap design or the bait. But using one trap model, that is the proma traps, in the Upper Zambezi Basin, we found out that the dog food bait had a significantly higher catch per unit effort than the cooked maize meal bait. Effectiveness of the dog food bait in attracting crayfish may be related to its high crude protein content, which is 180 grams per kilogram, compared to that of the cooked maize meal, which ranges between 50 to 84 grams per kilogram. Crayfish are considered to be highly sensitive to nitrogen-containing compounds such as those in meat-based dog food. Other high-protein baits, for example, liver, fish, chicken, and canned food, have been found to produce similar catch results as of the dog food. Dry dog food can then be useful as a standard bait for crayfish abundance studies in the region, as it is easy to handle, store, and requires less time to prepare for fieldwork. Other baits, for example fish, would need to be standardized according to the species, size, and probably condition of freshness, which would make the standardization process across the region cumbersome. There were no significant differences between the two trapping methods with respect to sex and carapace length of the Redclaw crayfish. There was a weak correlation between the catch per unit effort of the two trapping methods, and the slope of the trend was 2.68. This means that historical results obtained via opera traps baited with cooked maize meal should therefore be up-calculated by a factor of 2.68 for the data to be relevant and comparable across the region. From this study, we recommend the use of collapsible proma traps baited with dry dog food to trap a significant number of crayfish for management purposes, as well as a standard gear for Redclaw abundance studies in Africa. The method of crayfish population suppression as a management action might, however, not work in a huge system like Lake Kariba. This method can be used in smaller reservoirs and ponds to mitigate against potential impacts on native biota in invaded systems. It should also be noted that this method was only tested for the Redclaw crayfish and might not work for other species, for example, the European crayfish, spiny cheek crayfish, red swamp crayfish, and the marbled crayfish that all have established naturalized populations in African freshwater systems. Please feel free to ask me any questions or share any thoughts that you have.